So hopefully you went back and you tried to answer these questions, but I'll go over a few of the answers so you can make sure you're getting the right answers. But list and describe 3 structures common to all bacteria. So if you go back to this slide, you can see all of these: the cytoplasm, the chromosome, the ribosome, and the cytoplasm. So those are all structures that are common to all. And then these, any of these, the S layer, the fimbriae, the outer membrane cell wall, cytoskeleton of actin, pilus, capsule, inclusion, granule, microcompartments, plasmid or flagellum, endospore, intracellular membranes, nanowires, those are found in some. So that answers 2 of the questions. 3 general shapes of bacteria. So if you go back here, you can see coccus, rod, and curved or vibrio. And then 3 unusual shapes, or sorry, 2 arrangements found. And the arrangements would be like diplo is 2, tetrads 4, clusters or staph, chains or strep, sarcina are in 8s. And if you have the streptobacilli, they're end to end, and palisades are side by side. So that is a little bit from section 1. So we're going to start section 2 and the external structures. So we're going to go from the outside in, and you can see that the structure and function of the 6 different types of external structures, and then explain how the flagellum works in the presence of an attractant. Those are your learning outcomes. So the appendages, there's 2 major groups. We have flagella and axial filaments, which are internal flagella. They have lots of different names for that, but both of these provide motility. And then we have fimbriae and pili and nanowires, and these provide points of attachment or channels. So the flagella here has a hook, a filament, and a basal body that's embedded in the cell envelope, which includes the outer membrane in this picture. But it could just be the cell wall and cell membrane, or it could just be in the cytoplasmic membrane, depending on the type of bacteria. So it's not just found in gram-negative bacteria, which is, this is showing an outer membrane, so it would be a gram-negative bacteria. You can have flagella in gram-positive bacteria as well. It's just showing only, in this picture, is only showing the gram-negative. But you can see it's a very complex structure. It propels. It has an axis, a rod that it kind of wraps around, and it has a propelling kind of motion like a boat propeller. So the flagellar arrangements, we have polar flagella, and then we have peritrichous. Polar means that they're at one or both ends, and if it has mono, that means one single flagellum. If it says lopho, it's a tuft or a bunch at one end usually, or it could be at both ends, but lopho means tufts. And then amphi means both, so you have a flagella at both poles. And then peri means around or surround, and so this is like all over this the particular cell. So if we kind of look at these various examples that are in this, you can see if there's just the one, that's like A is the monotrichous, and the lophotrichous, you have a tuft, that's like the picture B down here. And if you have flagella at both ends, it's not really showing here, but that'd be sort of like C. And then D is the peritrichous, although it's really hard to see that. They're all around. E. coli is one that's a peritrichous. It has flagella all over, randomly dispersed all over the surface. Sort of like a bad hair day is the way I would kind of describe that. So chemotaxis and phototaxis. Taxis means directed movement. So as opposed to just random movement, this is directed by something. And you see that as the root word of photo and chemotaxis. So chemo is directed by chemical signals and photo is directed by light. So chemotaxis, you have positive or negative chemotaxis, which means that they are movement toward favorable stimulus, and negative chemotaxis would be movement away from a repellent or potentially harmful compound. So when you have positive chemotaxis, you typically have more runs than you do tumbles, and we'll show you what that means here in just a minute. And when you have negative chemotaxis, you have more tumbles rather than runs. And so a run is more of a sustained, smooth movement in one direction. And so let's look at that. So a run, it's counterclockwise movement, which seems weird. Counterclockwise means, you know, the opposite, like not the right direction. But, you know, like clockwise is the right direction because we see how clocks move, right? But in this case, counterclockwise is the movement of the flagella during a run, and it's in a smooth, linear direction, and it's going usually toward a stimulus. And where you have a tumble, they're reversing direction, and it causes it to stop and change course, and that's usually when you have repellents. So you can see here, if there is a tumble, it's kind of looking, and this is peritrichus right here. This is what peritrichus looks like. Has the flagella going in all directions, and so it's like, wah, bad hair day, and they're all spread out, and when they're all working together, so this stops them. When they are working together and they're all moving in this counterclockwise direction, then it's a run. And when it's going in the clockwise direction, it's a tumble, but that's more of a just kind of stopping the flagella. And so here you can see just stopping points are in red, that's a tumble. And where they're moving short distances, usually there's nothing attracting them, so we call this a random walk. But when they're moving in a longer distance and more of them are runs than tumbles, then they must be attracted to something and not repelled. Periplasmic flagella, also called axial filaments, also called endoflagellum, are found in spirochetes, and there are 2 or more long coiled threads that run the distance of the organism, and it's between the cell wall and the cell membrane. So it's internal, it's not external, and they give this flexing or twisting motion to the cell. So when you talk about the attachment or channel formation, there's a pili or pilus, and plural is pili. That's where you have adhesion, but not locomotion, and it's for sending DNA through from one cell to another. The fimbriae are also for adhesion, not for locomotion. And fimbria is singular, and fimbriae would be plural. So flagella can also be used for attachment in some species, but generally it's the pili and the fimbriae that do that. So fimbriae, little bristle-like structures. Composition varies in terms of the protein, but they're usually very short, and they may be responsible for formation of the biofilms. So this is an E. coli bacteria, and it has lots and lots of fimbriae to adhere to epithelial cells on the surface. Pili, you can see here, forming between these cells are passing genetic information, so it's in a process called conjugation. And so they've nicknamed that kind of bacterial sex. Even though bacteria do not do sex, they can exchange genetic information through the pili. And so it's made of a tubular protein, or it's a tubular structure made of a protein called pilin.